IgG is the most common type of antibody and makes up 80% of all antibodies. IgG molecules play a major role in the immune response by providing protection from toxins and pathogens. IgG provides protections through 3 different methods. The first method includes neutralizing pathogens by binding to key pathogen surface proteins and preventing the pathogen from interacting with the host cell. By blocking the pathogen, the antibody neutralizes the pathogen's ability to enter the host cell and replicate. The second method of protection includes attacking pathogens that replicate outside of the cell. To prevent pathogens from replicating outside of the cell, IgG binds and coats the surface of the pathogen and interacts with IgG-Fc receptors of phagocytic cells to mediate the ingestion of pathogens by the phagocytic cell. The third and last method of protection is through the involvement of the complement system. The complement system enhances the ability of the antibodies in phagocytic cells. In the classic pathway of complement activation, pathogens bound by IgG are recognized by C1q protein, a complement component, which triggers the complement cascade, which results in pathogen phagocytosis and lysis. IgA is the second most common type of antibody. Although IgA is the second most common type of antibody, it only makes up 13% of all antibodies. IgA's primary job is to defend against bacteria and viruses. However, IgA is also quite commonly associated with newborns. A mother's milk contains a high concentration of IgA that provides immune protection for newborns. Not only does breastfeeding decrease infant deaths from gastrointestinal issues, but also prevents respiratory infections. The IgA in the milk acts as almost a paint to prevent pathogens from adhering to different surfaces. IgM makes up 6% of all antibodies. IgM does not contain specific binding sites, unlike IgG, which allows B cells to produce IgM quickly during primary immune response. When an antigen is introduced into the body, large quantities of IgM are produced. Although IgG takes a greater role in removing antigens from the body, IgG is quite specific and therefore slow. However, IgM can bind to many antigens simultaneously and can quickly clear antigens from the bloodstream during the initial stages of an infection. Throughout the course of an infection, a rapid spike of IgM can be seen in the bloodstream, followed by a slow decline of IgM as IgG increases. Although IgM's primary role in the body is to be the initial response to infections, the antibody will react with antigens on red blood cell membranes after a mismatched blood transfusion. IgD makes up less than 1% of all antibodies. IgD functions primarily as an antigen receptor on B cells. When IgM and IgD expressed on a B cell interact with an antigen that they are specific to, the antigen is internalized and processed and presented to helper T cells, which trigger the B cells to proliferate and differentiate into plasma cells, thus initiating the development of the humoral immune response. Lastly, IgD is thought to play a role in allergic reactions. IgE makes up less than 1% of all antibodies. IgE's primary function is to promote inflammation and allergic responses. In an allergic reaction, antigens reintroduced into a previously synthesized individual binds to antigen-specific IgE on a mast cell and triggers release of pharmacologically active agents involved in immediate hypersensitivity syndromes.